Welcome to the Science Experience, your source for the most current and impactful research and preprints in science. Before we jump into today's deep dive, please take a quick moment to like this video and subscribe to the channel. It really helps support the Science Experience. Thanks. Okay, so today we're peeling back the layers on something really fundamental, how the levels of proteins in our cells are controlled. For a long time, we sort of thought we had the main story figured out, But new research is challenging that. It's pointing to a surprising factor that might be way more influential than we ever realized. Our mission today: give you a shortcut to understanding this vital process. It impacts everything: cell growth, tissue health, all that stuff. We'll aim for college-level insights, but keep it fun, accessible for everyone. Okay, let's unpack this. When we talk about how much of a protein is actually in a cell, it's always been seen as like a balance. And for years, the focus was squarely on the making side, how fast a cell synthesizes new proteins. That seemed to be the main driver.
Right, you've hit the nail on the head there. Protein concentrations are absolutely a dynamic thing. It's this balance between two main forces: synthesis, how fast they're made, and clearance, how fast they're removed. And yeah, the older models, they heavily leaned on synthesis being the primary regulator. The thinking was synthesis might only account for, maybe 5 to 8% of the variation across the whole proteome. Pretty small, really.
Only 5 to 8%.
Yeah.
Wow.
Yeah. And when we say clearance, it's important to remember it's not just about breaking proteins down, which we call degradation. It also includes something less obvious, but super important: dilution. How proteins get sort of spread out as a cell grows and divides.
Dilution. Right.
Exactly. If the cell gets bigger or splits, the existing proteins are suddenly in a larger volume, so the concentration goes down, even if none were actually broken down.
Okay, that makes sense. Like juice in a bigger glass.
Precisely.
Yeah.
So if synthesis was the star for so long, this new research must be asking, well, does the growth rate of the cell change the whole game?
Does how fast a cell grows shift that balance between making and clearing proteins?
That's exactly the core question, and the answer seems to be a pretty resounding yes. The relative importance of degradation versus synthesis, it turns out it's really heavily dependent on how fast that cell is growing. Think of it like this. In slow-growing cells, protein levels are mostly dictated by how fast they degrade. Slow degradation means they build up.
Okay.
But in rapidly growing cells, that build-up effect gets dampened significantly because the proteins are diluted much faster than they break down. The growth itself, the expansion, kind of overwhelms the degradation rate.
Right. And this is where it gets really interesting, isn't it? The actual numbers they found.
Yeah.
They looked at a ton of proteins.
They really did. Over 12,000 proteins across different human cells, mouse tissues, using some very advanced techniques like plexDIA, super precise stuff. And the results are, striking. They found that growth-dependent dilution by itself, just that spreading out effect, can explain about 40% of the changes in protein concentration. 40%, just from dilution.
40%. That alone is huge.
It really is. And here's where the old view really takes a hit. In slowly growing cells, think natural killer cells or brain tissue, the variation in degradation rates can explain, get this, up to 50%, sometimes even hitting 62% in specific cases, of the variation in protein amounts.
50, even 60%. That's massive compared to the old 5-8%.
Exactly. It's substantially larger than previously appreciated, as the paper puts it. Degradation is a major player in slow growth. But then, flip the coin. Look at rapidly growing cells. Fibroblasts, bone marrow cells.
Yeah.
Their degradation accounts for only about 7% of the variation. Synthesis explains the other 93%. It completely flips.
Wow. So it's totally context dependent.
Totally. Growth rate really dictates who's in charge: synthesis or degradation.
Okay, that leaky bucket analogy really helps here. Slow filling, the leak size degradation matters most.
Mm-hmm.
But if you're pouring water in super fast or the bucket itself is rapidly expanding like cell growth, that leak suddenly becomes way less important. The filling or the expansion takes over.
That's a perfect way to visualize it. It captures that shifting control beautifully.
So conceptually, that makes sense.
Yeah.
But how does it actually play out in, a real biological system? Let's zoom in on those B cells you mentioned, immune system powerhouses.
Yeah, the B cell model was great for this, because you can have them resting, not dividing or activated, where they divide like crazy, maybe every 16 hours. Huge growth rate difference.
16 hours.
Right. And what they saw was this thing called incomplete compensation. When the cells ramp up growth, they have to make more protein, right?
Makes sense.
But the study found that synthesis increase doesn't always fully match the dilution from that super fast growth. The change in clearance, mostly from dilution, actually explained 37% of the protein concentration differences between the fast-growing activated cells and the resting ones.
So nearly 40% of the changes were just stuff getting spread thin.
Pretty much. Faster than they could top it up in many cases.
And what does that mean functionally, like for energy production? You mentioned that.
Great question. Proteins for the electron transport chain, absolutely vital for efficient energy production. Their concentration dropped by half, twofold, upon B-cell activation.
A half. That's a big drop.
It is. And it was mostly driven by dilution. It reflects a known metabolic switch. Activated B cells shift from efficient energy production, oxidative phosphorylation, to a faster, less efficient method, glycolysis. They prioritize speed for division.
Ah, okay. Speed over efficiency.
Exactly. But, and this is important, not all proteins just get diluted away. Some critical ones, like histones, the proteins that package DNA, they defy the trend.
They stay constant.
Pretty much. Their synthesis rates are actively boosted to perfectly compensate for dilution. So the cell can override this effect for things it absolutely needs to maintain. It shows targeted control.
That's fascinating. It's not just a passive process then. So this isn't just in isolated cells, right? You said it shapes whole tissues. How did they show that in, like, a whole mouse?
Right, they looked in vivo. They compared mouse tissues with vastly different growth rates. Brain tissue, super slow turnover, maybe doubling time up to 200 days.
200 days.
Yeah, very slow. Compared to bone marrow, one of the fastest, doubling time around 1 day.
Huge difference.
Massive. And the findings, they mirrored the cell culture results almost perfectly. In the brain. Slow growth. Degradation explained about 50% of protein abundance variation.
50% again?
Yep. But in the fast-growing bone marrow, clearance rates, degradation plus dilution had a much weaker link to protein levels. Dilution just dominates. They saw this continuous trend across all tissues, a really strong correlation, like 0.89, between growth rate and how much degradation mattered.
Almost a perfect relationship. So what's the bottom line for tissue identity? How does this make a brain different from bone marrow at the protein level?
It's fundamental. When you have that big growth rate difference, brain versus bone marrow, the shared differences in degradation rates, which are heavily influenced by dilution in the faster tissue, drive the protein concentration differences. It explained about 34% of the changes between brain and bone marrow.
A third of the difference just from this growth clearance link.
Exactly. But if the growth rates are more similar, say brain versus pancreas or lung, then the specific degradation rates of individual proteins become more important in setting the differences. It's more about protein A being less stable in the pancreas than the brain, for example.
So it depends on the scale of the growth difference.
Precisely. And the overall impact is huge. For many individual proteins, their level across tissues is almost entirely explained by how fast they're cleared. Some have correlations over 0.7.
70% explained by clearance alone.
For certain proteins, yes. Think about pathways important for the brain, like Neurotrophilin signaling or WNT5A signaling. Those proteins are more abundant in the brain, mainly because they degrade much more slowly there.
Okay. My mind is slightly blown already. But then you mentioned another layer. Different versions of the same protein.
Ah, yes. Proteoforms. This is super cool. So one gene doesn't always just make one protein.
Right, alternative splicing and stuff.
Exactly. Alternative splicing can create different versions by mixing and matching parts of the RNA. Or sometimes even tiny changes, like one amino acid substitution from alternate decoding. These are all proteoforms, different protein products from the same gene.
And degradation affects these differently, too.
Significantly. In the brain, they looked at about 500 alternative splicing events. For 34% of them, the different proteoforms produced had significantly different degradation rates.
A third of them?
Yep. For instance, one version of microtubule actin cross-linking factor 1 degraded faster, and guess what? It was less abundant. Same for gelsilin proteoforms. Faster degradation meant lower levels, a really strong link.
Wow.
Even single amino acid changes mattered. One substitution in EPS15 made it more stable, so it was more abundant. Another in PLP1 made it less stable, decreasing its abundance.
So this explains why, even if a gene could make lots of different versions, we often only see a few, because some are just less stable.
That's exactly the implication. It provides a direct molecular reason. Protein stability, driven by degradation, acts like a quality control or a selection filter. It helps determine which proteoforms actually stick around and function in the cell beyond just how much RNA is made. It's a fundamental layer of regulation.
Okay, let's try and wrap this up. Key takeaways for you listening. First, Cell growth rate. It's critical.
Boom.
Way more important than we thought for deciding how much impact protein breakdown has.
Absolutely critical.
Second, in those slow-growing cells, degradation is a huge driver of protein levels, explaining maybe up to 50% of the variation, much bigger than the old model suggested. Third, differences in clearance, that mix of breakdown and dilution, are fundamental in shaping the unique protein landscapes of different tissues, brain versus bone marrow, largely down to this. And finally, the really surprising bit, maybe.
Yeah.
Degradation even fine-tunes the levels of different proteoforms, different versions coming from the same gene.
And we should stress these conclusions are built on really solid, rigorous work. Multiple methods, advanced tech. It really challenges that old synthesis-centric view. It shifts the balance towards clearance, especially considering growth rate.
It definitely does.
And it leaves us with a really interesting thought to chew on. If long-lived proteins accumulate more in slow growth states, does that mean they're generally less needed or maybe even get in the way during high growth, but become crucial for low growth states? Think about what that might mean for understanding conditions where cell growth is altered, things like aging or diseases like cancer. Could this shift how we think about targeting those processes?
That is a fantastic, provocative thought. A lot to think about there. Thank you for joining us on this deep dive into protein regulation. And one last reminder, please do like this video and subscribe to the channel. It genuinely helps us keep bringing you these explorations. Thanks again for tuning into the Sand Science Experience. We'll see you next time.